Good morning, everyone. Today we are discussing rewiring the cardiac circuit. We will explore how the heart decides between adaptive growth and destructive remodeling. We are moving beyond simple symptom management toward a future where we use systems biology and RNA therapeutics to fundamentally rewrite the heart's fate. When the heart faces mechanical stress, it must make a critical biological decision based on Laplace law. It increases mass to reduce ventricular wall stress. Physiological adaptation is triggered by endurance training or pregnancy. This results in eccentric growth, proportional chamber enlargement that preserves the function. Pathological remodeling is triggered by hypertension or infarction. This leads to concentric growth, severe wall thickening, and eventually contractile dysfunction. The initial stimulus determines if the long-term result is protective or destructive. Physiological versus pathological growth. How do we distinguish these two stages clinically? Physiological growth is reversible post-stimulus, while pathological growth progresses stubbornly toward heart failure. Pathological growth is marked by severe interstitial fibrosis and the reactivation of the fetal gene program. In physiological growth, angiogenesis keeps pace with muscle growth. In pathology, insufficient capillary density leads to hypoxia. At the molecular level, the heart's fate is decided by two competing networks. The adaptive network, driven by insulin, IGF-1, nitric oxide, and thyroid hormone. This pathway ensures preserved function and cell survival. In the maladaptive network, fueled by angiotensin II, endothelin 1, and catecholamines, this pathway leads to directly remodeling and failure. Our therapeutic goal is to shift the signal from path B to path A. This flowchart explains the execution of the maladaptive circuit. In receptor activation, angiotensin II binds to GPCRs. The calcium influx sustained stimulation releases a massive flood of intracellular calcium 2+. The molecular switch activates calcineurin, a calcium-dependent phosphatase. In nuclear execution, calcineurin dephosphorylates NF-80, which moves into the nucleus to transcribe fetal and pro-hypertrophic genes. In contrast, the adaptive circuit uses receptor tyrosine kinase. In kinase cascade, PL3K recruits and activates AKT. In cellular coordination, AKT activates mTORC1 for controlled protein synthesis, while Hippo-YAP and VEGF pathways ensure capillary growth matches myocyte size, preventing the hypoxia we see in diseased hearts. We must also view the heart as an engine. In healthy adult engine relies on efficient high fatty acid oxidation for high mitochondrial capacity. In failing pathological engine undergoes a fatal shift to glycolysis and ketone reliance. This leads to an energy deficit, lipotoxicity, and ceramide accumulation, which ultimately triggers apoptosis. Pathology also unlocks forbidden parts of the genome. In histone modifiers, the class 2 HDACs normally act as repressors. Oxidative stress forces them out of the nucleus, removing the padlock from pathological genes. In chromatin remodeling, SNF complexes physically expose hidden fetal genes. The RNA interference, long non-coding RNAs like chair block the silencing ability of the PRC2 complex, allowing pro-hypertrophic transcription. MicroRNAs are our most precise silencers. These 22 nucleotide strands incorporate into the RISC complex. Their seed sequence fits perfectly into the 3' UTR of target mRNA, resulting in immediate degradation or severe translational inhibition of entire disease networks. The current ongoing research testing three major RNA inhibitors in the clinic. Ladomycin targets MIR21-5P in macrophages to stop fibrosis. MRG110 targets MIR92A3P to promote angiogenesis. CDR132L targets MIR132-3P, a primary driver of cardiac stress and remodeling. To find these targets, we use high throughput data and computational modeling. We analyze thousands of in vivo single gene knockouts from databases like IMPC. We then map neighbor genes connected to no hypertrophic hubs and validate them in vitro using siRNA knockdown in neonatal rat cardiomyocytes. Our systems biology approach has uncovered 5 novel modulators. The brakes, negative regulators, genes like LRI-G1 and CBL naturally suppress disease. Knocking them down increases hypertrophy. The accelerators, positive regulators, the genes like VAV2, LEPR and RASA1 drive the disease. Knocking them down decreases the hypertrophic phenotype, making them promising drug targets. We cannot ignore the cellular neighborhood. The cardiomyocyte is constantly receiving signals. Fibroblasts secrete TGF-beta and exosomes to drive hypertrophy. Endothelial cells provide protective nitric oxide or stress-dependent paracrine regulators. Macrophages balance inflammatory destruction with repair signals like interleukin 10. This is our master map for the future of treatment. We are moving toward a combination approach. In receptor blockers, cutting off the GPCR nodes. In metabolic modulators, using SGLT2 inhibitors to shift the energy dials. In epigenetic and RNA modulators, overriding chromatin paralogs and silencing destructive RNA sequences. In novel system targets, plugging in hubs like LRIG1 and VAV2 for future combinations. In conclusion, we are witnessing 3 major shifts. First one, from single node to network, realizing that silencing one receptor is not enough. The second one, harnessing RNA, using oligonucleotide for genome level precision. The third one, orchestration, repairing the mitochondrial engine and the non-myocyte microenvironment is just as vital as stopping the initial trigger. Thank you for joining in this lecture.